Okay, perfect. Okay, well, welcome everybody to another MCRN Colloquium. It's nice to be back after a couple weeks without any talks. So today we're lucky to have Dr. Laubmeier, Amanda Laubmeier. She's an assistant professor in the Department of Mathematics and Statistics at Texas Tech University. Her research sits at the intersection of mathematics and ecology. She focuses on the mathematical study of ecological systems. Before joining Texas Tech, she was a postdoctoral fellow. I think held the Marilyn Hitz postdoctoral faculty fellow position at the University of Nebraska-Lincoln. And before that, she got her PhD in applied mathematics at NC State in North Carolina. So today, she'll be talking to us about modeling biological control services in agricultural ecosystems. And with that, I'm looking forward to the talk. Thanks, Amanda.
Thank you for having me. I'm excited to talk in the colloquium with the caveat that I don't do like a lot of climate research, so I try to connect as much to the topic, but it's going to be maybe a loose connection. Instead, the type of thing that I tend to think about is biological control. So I've been getting a lot of mileage out of this comic I saw online earlier in the year about a gardener looking over some tomatoes and seeing a little ladybug who's going to provide some biological control services. And I like the last panel because it shows how grisly a process it can actually be. So this interaction is the type of thing I like to model, and I like to figure out like how happy we can make that gardener. So sort of my motivation for studying this type of thing and just general approach is that I think as mathematicians, we all know that math can provide insight into factors that drive ecological change. And models are especially helpful because they allow us to explore ecosystems under novel conditions, which is valuable in the context of climate change because we don't know what conditions an ecosystem will face in the future. I'm especially interested in insect pests in agricultural fields. And when I think about those pests, I also think about the community of insects that are their natural enemies that perform biological control services. And insects are really sensitive to temperature. So we expect that their interactions are going to change with environmental conditions. The types of communities that we see and the types of feeding interactions that they show, we expect to be dependent on the environment. So during this talk, I decided to focus on two different types of projects where I thought about this overall question, but sort of in two different contexts. The first, I thought about how insects respond to temperature. It's older work for me, but I thought it was the most related thing I had to this sort of colloquium's overall theme. And then the second half, I'm going to talk about how insects respond to landscape and pesticide use, kind of more directly how people interact with these systems. And although it's not related to climate, I think it's related to the same types of things that we think about, like how people interact with systems and how their interactions might change in the future. But getting into that first topic, I'm just going to be talking about predator community composition and how it responds to and relates to temperature. So I'm thinking about biological control of aphid pests. An aphid is this little green thing, and a possible biological control agent is the spider or like the ladybug I showed in the comic on the first slide. So aphids are a widespread pest, but they're also the natural prey of many different types of ground beetles and spiders. Sort of our intuition is that if we have more of these different types of predator species present in an agricultural field, then we'd have improved biological control services by the predator community. To build intuition for that, you think about if you have 10 different types of predator and they're active at different times of day, like some are in the morning and some are at night, then you would need to collect predators from both those categories in order to be controlling the pests throughout the day. So the type of question that I'm wondering about here is which species would provide the most pest control of the aphid, and what types of predator diversity, like what ways that they respond to their environment, are going to improve pest control. There are some complicating factors that make this like an interesting modeling question. So the predators that I'm thinking about are generalists and consume a range of potential prey items. So here's a picture of a spider and it's eating another spider instead of eating the aphid I showed you before. The ladybug on my very first slide is actually a specialist, so it's not something I'm thinking about in this particular project. Instead, I'm thinking about things that eat lots of different foods. And so the reason this matters is because as we increase the number of other predators in this community, we're also increasing alternative prey for those predators or things that might eat them in turn. And then as I consider all these different predators, it's important to bear in mind that their behavior and life cycle is going to be strongly driven by temperature. So we're sort of wondering, as these communities of predators become more complex, would we expect pest control to be improved or impaired? And then once we have a handle on that, we'd ask, if temperature changes, how would that change the efficiency of this community that I'm thinking about? So I have worked on this model for a while that sort of describes predator-prey interactions in terms of different traits. And so here I'm just going to give you an overview of the different types of behaviors that are important in this model and kind of why we include them. So attack rates, handling times, and death rates, I'm going to describe with a body mass dependence. So we're kind of thinking that large species are going to be highly mobile. They have longer legs, they cover more ground in a step, and so they're more likely to encounter other individuals. Also, predators are well suited to consuming a particular size of prey item. So that's shown kind of in this picture here, where along the x-axis is the size of potential prey, and along the y-axis is how likely they are to eat it. And the centipede right here, this dark green curve, it's not so likely to eat the spider. It's too large for its preference, but it's more likely to eat this little green bug because it's closer to its preferred body size to eat. And then another way that body size matters is because large species have higher metabolic demands, and something that's kind of interesting is that those metabolic demands are likely to increase with temperature. So there's this trade-off that we're going to see where large size can increase attack rates on the prey, but it's also going to increase intra-guild interactions that can sort of reduce pest control, and it's going to increase metabolic decline. So larger predators are going to sort of suffer at higher temperatures. Another thing that we think about in this model is how encounter rates depend on overlap in foraging activity. So we assume that predators forage over some area for prey items. That's illustrated in this diagram here, where these three different spiders are predators and they have different parts of the plant height that they like to occupy. This is sort of a ground-dwelling spider, and this one, I guess, builds webs up in the leaves. And so in order for one of these spiders to eat the grasshopper, its preferred area necessarily has to overlap with the grasshopper's area, which is the entire height of the plant. But we might also have that the spider's habitat areas overlap, like where these ovals overlap with each other. And when that overlap happens, or how much that overlap happens, is going to determine whether there are encounter rates between the predators, which can lead to negative intra-guild interactions, like consumption of a smaller predator by a larger predator, or just competition. So again, we're going to have a trade-off here where if a predator has a larger foraging area, it has more chance to encounter the aphid and eat it. So it might improve pest control, but also it increases the opportunities for intra-guild predation, so it decreases pest control. And so all this is built into a model that it's just an ordinary differential equation describing the density n sub I of species I changing in time. And my time is going to be restricted to a single agricultural growing season where my pests, my aphids, experience population level growth. They reproduce quite quickly. And my predators, they decline at some rate x sub I because they can die off over the course of the season, but they actually experience like seasonal breeding. So I'm not accounting for their growth rate here. Additionally, the second term right here is a big Lotka-Volterra type attack rate term, where I sum up overall potential consumers of whatever species I'm currently thinking about, species I. And then there are attack rates at some rate, nu ij, aij, where nu is going to describe those encounter rates that depend on space, and aij is going to describe those attack rates that depend on body size and feeding preference. And then just as either species is more abundant, as long as they overlap, they'll have more attack rates. This denominator is just a big functional response that reduces attack rates according to the time that a predator spends engaging in other activities. The first term right here is sort of like a standard Hulling type 2 functional response, where I'm accounting for the time a predator spends consuming other prey. So I'm looking at the potential attack rate on some different prey item of species K, how long it would take to handle that prey item, and that handling time is again going to depend on the size of my predator and the size of the prey item it's trying to process, and then abundance of that alternative prey. So as I see more of this prey, especially if it's very likely to overlap with my predator, then I'm going to see more time spent on this type of activity. The second term is kind of like the competition terms you would have in like a Beddington to Angeles functional response, but this is a little bit more generic because it's accounting for the entire community of predators. And sort of what we're thinking this term should do is it describes the time that a predator spends evading its own predators, with the understanding that the species in my system are generalists, so pretty much they're happy to eat anything else in the system. And what I'm going to do is I'm going to sum up over species L that might potentially eat the predator that I'm thinking about. And then as long as the two predators overlap with each other, then if there's more of this larger predator, it's going to decrease the time that the smaller predator has to eat its prey because it has to evade that consumption event. Just as a reminder, temperature is going to affect these encounter terms. It's going to, in the first version of the model I show you, just change the death rates and growth rates, but in the second version, we think about how sort of habitat can change with temperature as well. And then all of the parameters are formulated in terms of species characteristics, like body size. And in prior work, I had actually parameterized this type of model to field data and lab data to see how well it performed. And I used the scaling constants from that parameterization to run this whole thing. What I'm going to do is I'm going to apply my model to a case study where I'm thinking about a bunch of beetles and spiders in barley fields. So I consider the problem of controlling an aphid, and I'm thinking about a particular aphid, our Patty, using a community of spider and ground beetle predators. So I pulled this community data from a project I'd worked on previously where we had parameterized this type of model, but now I'm sort of thinking about this predator community and how it would respond in different ways. So this diagram shows you the community I'm thinking about. I'm just going to describe some features of it to you real quick so that when I talk about these predators, you kind of know what they represent. The left axis, the dark bars, are representing the size of my predators. So I have some very large and also some very small ones. And then the right size, the light bars, are representing the average density of these predators in the field. I don't use this a ton in this work because I'm thinking about something a little bit more like theoretical, but just so that we know, like, some of these predators really are abundant, whereas others, we don't see that much. And then this left half are ground beetles and this right half are spiders. And when I start thinking about where these predators are likely to reside, I'm going to rely on that distinction to sort of treat them differently. So the approach here is something that I started working on during my postdoc at the University of Nebraska-Lincoln with Brigitte Tenenberg in biological sciences and Richard Rebarber in mathematics. And what we decided to do was use this ODE model that I had previously parameterized, and I had cool field data to connect it to. And we knew that it described aphid consumption, which we wanted to track. How well can we get rid of the pest, as well as a bunch of cool features like intraguild predation and interference. Our model parameters emphasize how predators respond to temperature and what sort of characteristics they have, like how big they are or how large an area they can cover. And so we can use it to explore how those different features are related. And what we're going to do is sort of, since we have an ODE model and it's been parameterized, if we have an initial distribution of predators, then we can forward solve our model to get the abundance of the pest over a growing season. And then what we want to do is solve an optimization problem where we identify the initial distribution of predators, the initial condition for my model to plug in, that's going to minimize the average daily aphid abundance. So I have a problem of trying to control the pest as well as I can. So I think the metric for that is average daily abundance. And then I have a feature that I'm allowed to change. That's going to be how my predator community is set up. Sort of in an ideal world, imagine that I could pick which insects are in this field. How would I pick them to minimize the pest density? And in doing that, we're just going to explore how different characteristics of the predators or the effective temperatures on those predators changes their performance as a biological control community. So the first thing we did was sort of see how temperature and body size play off each other. So what I have in this diagram, and I'm going to show you another one similar to it in a second, my horizontal axis is temperature ranging from 15 degrees Celsius to 45 degrees Celsius. I'm just going to think about fixed temperatures for the time being. So for the entire simulation I run, I'm at 15 degrees. And then up and down on this graph vertically are the different predators I showed you before. They're arranged from largest to smallest, but separated again by those ground beetles and spiders. So these are my beetles, and down here is my largest beetle. And then these are my spiders, and up here is my smallest spider. And then each grid in this figure is showing me what percent of the optimal predator community that predator occupies at that temperature. So if I take a vertical slice, I'm going to be looking at the optimal predator community at like 25 degrees. And then for every predator, I check how much of the community I should devote to that predator in order to optimally control the aphid. And so in the 25-degree example, I should put everything into this one beetle. Some caveat for these results are that we're ignoring foraging areas. So we're assuming everything is occupying the same space, kind of the standard way that we set up, like an ordinary differential equation model for predator-prey interactions. And we're also considering those fixed temperatures where every vertical slice, the temperature is always the same. The general trend we're seeing is that at lower temperatures, the large predators are more efficient because they move more, they can eat more. But then as temperature increases, smaller predators become more efficient because of that trade-off I mentioned between movement and metabolic demands. Kind of as it warms up, these predators need to eat more to survive, so they kind of become less valuable in the community. Next, we thought about sort of making it a little bit more complicated, where we change how foraging area is determined for the different predators. We decided to set predator foraging area based on classification as a beetle or a spider. So a species is going to overlap very strongly with members of its own species. They all use the same area. But then it's going to have slightly lower overlap with the aphid prey or predators within the same category. That is beetles with beetles or spiders with spiders. And then the very lowest overlap is going to be between beetles and spiders. And so what's cool is that compared to the earlier results, we see that more than one predator is in our optimal predator community, which is kind of what we expected to see. We still have that general trend that as we move from left to right, we move from our largest to our smallest predators. But we see this gradual shift where we sort of hold on to more than one group of predators as we go along. And we have both the spiders and the beetles kind of in the same optimal community. And there are a couple sudden jumps we see. But for the most part, we can kind of break it into 3 regions of separate behavior. So that was kind of part 1 of this project, but in the field, temperatures aren't constant. So we thought that these results weren't super realistic, and maybe we might see more changes if temperature was allowed to vary. So we decided to look at habitable temperatures changing foraging activity instead of those very simple assumptions we made before. And so this is something I worked with a PhD student when I first got here to Texas Tech. She's since graduated and now she's a postdoc at Virginia Tech. And what she did was she generated these curves where along the horizontal axis is temperature and then along the vertical axis is attack rate on the pest. And the peak of these curves is sort of the temperature at which these predators are the most active. And these sudden drop-offs where they kind of sharply go to zero is where it's too hot for the predator to continue to be active. And in the top panel are my larger predators, and in the bottom panel are my smaller predators. And then the kind of blue-toned solid lines are my beetles, and then the more red-toned dashed lines are my spiders. I've just broken it up this way because the magnitude of the attack rates is quite different between my large and small predators. But the temperatures that they use can kind of I wanted to make sure that you could see the level of overlap that you have between the beetle and spider communities when we think about that smooth change in temperature. So when we combine those curves with field temperatures, we can see the combined effect of habitat use and temperature on attack rates on the aphid. So this bottom panel right here is temperature. I'm just looking at a snapshot over 48 hours. And this temperature was pulled from the same data set as I pulled my predator community. We had temperature loggers in the field that recorded the temperature every like 15 minutes. So I'm just manually plugging those into my model and solving over the field season. And so as temperature fluctuates, I'm showing you the attack rate by one of my spiders and one of my beetles. And then sort of what we're seeing is that at high temperatures, predator activity is going to drop. Kind of they need to seek shelter because it's too hot to continue. So you see these flat parts of the attack rate for the beetle. At that temperature, it just needs to seek shelter. It's too hot. But that temperature is not too hot for the spider, for example. And then the difference between the solid and the dashed lines is to show the effect of the predator functional response. So the solid line is the possible attack rate that the predator could have on the aphid at that temperature. And then the dashed line is the effect that you would have if you account for the functional response with an average community of predators and aphid, sort of reducing the time that it has available to hunt the pest based on the handling time or evasion time it spends with other predators or prey items. And so sort of the reason I think this is cool is because we see that the times when the predators are most active are also the times when we see the greatest loss in effective attack rates due to the functional response. Kind of they're more active, so having more interactions, so there's more likelihood of these handling times or interference rates reducing the attack rate. Whereas when the attack rate is lower, it sort of has less of an effect. And so we took those attack rate curves and we just tried to optimize the initial predator community in order to reduce the aphid pest population. We had 10 different fields where temperature and predator data had been collected. So we just solved this optimization problem for 10 different model simulations that are driven by the temperature. And each of these dots represents sort of the percent of the optimal community for a single temperature time series for my different predators along the horizontal axis. And the two different panels are just that this is like 0 to 100%, but then I zoomed into the lower 15% so you can see a little bit of what's happening with the lowest performing predators. The horizontal lines are going to be the average between all the different temperatures. And then the star is actually, if I try to optimize over all 10 temperature time series simultaneously, what's the optimal predator community? And kind of what we found wasn't what we wanted to find. We were kind of hoping we would see more diversity in the optimal predator community, but that's okay. We saw that Lycosidae was the optimal predator in most fields. And then this other carabid group, which is kind of a bunch of species that just couldn't be grouped with other ones, they also benefited pest control. And we found that the true predator community did not control the aphid population, like when we forward solved the model. But these optimal ones control the aphid population in 8 out of 10 of the fields. So they are performing as well as they can. Aphids grow quite quickly, so it really is hard for a predator community to control them. And then when we optimized overall simultaneously, we did see that shifted us even more towards prioritizing only Lycosidae compared to taking the average across all fields, where we see some value to variability, comparing the distance between this horizontal line and that star marker. Something else that we were able to do with the way our model was set up was we could kind of look at how temperature is affecting the attack rates on the pest community, on the pest population. So this is for the optimal predator community. And we're just looking at how the attack rates change with or without temperature dependent habitat use. So that's going to be the difference between my blue and black lines. So the black lines are horizontal because they don't change with temperature. And the blue lines have this hump-shaped curve that I showed you for how predators respond to temperature. And then I'm looking at with or without that functional response, or those intra-guild interactions, with my solid versus dashed lines. So thinking about the non-temperature dependent rates, this top line is sort of the possible attack rate on the aphid. And then this dashed line is once I account for handling times or interference. And same for this blue one, where I do account for temperature. And then this is accounting for temperature and handling times or interference. And what's cool is that kind of as we'd expect, the temperature dependent attack rate curves are below the curve where I don't account for the fact that they seek shelter sometimes. And my attack rates with functional response are lower than my attack rates without functional response, as you'd expect. But there's this really cool region right here where if I account for temperature dependent activity and functional response, I see higher attack rates than when I only consider temperature, or only consider my functional response without temperature dependent activity. And this is where we're seeing the benefit of the predators using their environment in different ways, kind of temporal partitioning of the pest control based on just when they are physically active. And so it's a small difference, but it is sort of important to consider both those things. We also tried simulating increased temperatures, where we just took our temperature series and then we shifted them upwards, accounting for an average increase in temperature, and also stretched them out, sort of making temperatures more variable. And we just saw way less variability in optimal communities. And it also shifted to sometimes Lycosidae was no longer the most beneficial, but a different small spider. And we lost a lot of control. So in the case where the temperatures are increased, the communities could only control the pest in 4 fields compared to before we had control in 8. So just some takeaways from this project were that we were able to optimize a trait-based differential equation to try and understand what characteristics make a predator and its community effective in some agricultural fields. The way we set up the model based on these mechanisms we wanted to study helped us to test how different predator interactions can increase or decrease pest control. And then we were able to simulate how temperature sort of changed and ultimately reduced the effectiveness of our predator communities. I do want to include the caveat that it took years to develop and parameterize this model, and it's still missing a lot of important features. So there could be additional work like improving the model, but at a point it just becomes very complicated to figure out appropriate parameters. With the rest of my time, I had planned to be done in 45 minutes based on what I saw on the website, so this project's a little bit shorter. But I want to talk about landscape and management. This is the project that's not related to climate change, but I think uses similar ideas. So I want to call back this kind of diagram I showed you before. And we often assume that population models have these uniform habitats with well-mixed populations. But I argued before that predators hunt and seek shelter in diverse ways, leading to different pest control outcomes. Why this matters in agricultural ecosystems is that the most hospitable places for predators may not necessarily be the places where pests reside. We're going to have these huge, clear-cut, dirt areas with the crops that the pests sit on, and then surrounding it untreated areas where the predators might tend to be. So we really need to understand where predators reside in order to assess the effect on pest control services. And here's a picture of kind of a landscape that includes agricultural fields, just to sort of show how agricultural fields can come in different sizes and are part of a really complex landscape. They can be surrounded by like urban areas or natural vegetation, or in some like parts of the US, just lots and lots of other agriculture. Within a field, managers can take different actions in order to sort of encourage a predator presence, and that might affect the pest control that we see. And so the concept for this project came to me from John Banks, who's an entomologist at Cal State Monterey Bay. And so in this project, I'm modeling predator movement. And so I switched to a partial differential equation based on something by Arditi in 2001. And so the general approach is that I'm incorporating some important spatial processes and interactions. I found some model parameters that match the empirical literature. Parameterizing from data wasn't really feasible for this model. And then to solve it, I did it in probably the silliest way possible because I am not a numerical person. I discretized spatial derivatives into a big system of ODEs, and I solved that. And again, I only simulate one growing season, and I just change model parameters in a way to see what the effect of common management choices will be. To show you the model that I'm thinking about, it's this system of partial differential equations, giving the density of the pest, my aphid, A, a predator, B, and then the directed movement of my predator, V, which I'll talk a little bit about when I get to that equation, and just how they develop in time. And then these states, A, B, and V, are going to have positions at every point, or values at every point in space, which can be 2D or 1D. So I have logistic growth of my pest, which doesn't depend on space, as well as the fact that if a pest and predator are in the same location, then a feeding interaction can occur. The pest might be eaten by the predator. I'm thinking about a really simple, like one predator, one pest community here. So I've stopped thinking about those really complex intragilled interactions. This is kind of more inspired by that specialist predator case. And then the movement for the pests, the aphids, is that they diffuse due to crowding. For the most part, they kind of asexually reproduce and stay on the same plant. But if they start to bump into each other a lot, they trigger a mode of reproduction that causes them to fly to new plants. And then what's kind of cool is that predator movement on a plant can actually accelerate that rate of diffusion. For the predators, I have diffusion at some rate that sort of describes just their randomly foraging in the field over the course of a day, and then also directed movement at rate V. And that's where the state V comes in. I describe the development of their directed movement with this differential equation, where they follow increasing prey density. So their velocity is positioned towards the prey. They exhibit prey taxis, but that movement is smoothed out due to competition. So if all of the predators try to shift in the same direction at once, if this V is very strongly aligned in one direction, they're less likely to all be able to move that way. Instead, they might scatter out. And then predators can be introduced to the middle of the field at some rate by managers. Or they might aerially migrate in, but that's kind of less likely. So when I set this up as a 2D system of equations, I can sort of create a heat map of abundance. I have snapshots on day 0, 30, 60, and 90 of my growing season. And this is just showing the heat map is of aphid abundance. The different snapshots are how it develops in time. And then this is the case where the predators are introduced to the field's interior. So I just start with like random noise for the aphids, and then you start to see these kind of bubbly patterns of the predators kind of moving in and chasing them around the field. We have boundary conditions, and I'm mentioning them now because that also is a way that predators can enter the field. So for the most part, I'm assuming a closed field. The pests don't really have anywhere to go outside the field. And the predator directed movement, because of that, wouldn't be toward the field edge. And their directed movement isn't going to change across the edge. But the predators, if there's natural vegetation at the boundaries of the field, then they might be encouraged to migrate into the field at some rate that's given here. And when I consider that case, I have the sort of same setup of heat maps, but now instead of sort of aphids spread around the field, they're really centered in the middle because my predators are just coming in from the edges and consuming the pest population there. I'm also seeing much higher pest densities. It was quite low before. The predators were very effective at biological control, but in this case, they're quite ineffective for that middle region. And so we kind of saw when we ran simulations for differently sized fields that as the field size in hectares increased, then the aphid density, the pest density increased dramatically. But it did saturate for the largest fields. Kind of after a point, the field is so large and the predators are so ineffective that further increasing the size won't do anything. And here you're thinking about like American conventional agriculture. Down here would be your smaller like fields, maybe more like you see sometimes in Europe. And then we decided to modify the model to incorporate pesticide effects because usually biological control agents are used alongside traditional pesticide sprays instead of by themselves. But even when a pesticide is targeted towards the pest and not lethal to natural predators, it can have these sublethal negative effects on their efficiency or movement. So we sort of added a class of predator that's been affected by the pesticide, and we added equations to describe their movement, but it followed the same rules. It was just reduced due to pesticide effects. So the reduced parameters were the directness towards prey. They kind of are less effective at sensing it, or their random movement, how fast they forage. And we just implemented the pesticide as whenever the aphids exceeded a threshold, the entire field was sprayed and predators had some chance of being affected. What we saw was that aphid density increased as predators became less mobile because of pesticide exposure, which is what you'd expect from the way the model is set up. In these graphs, what I'm showing you along the horizontal axis is how fast a predator was before it was affected by the spray. And then along the vertical axis, I'm showing you how much the aphid density increases when they experience the effects of the spray. And I have the two different panels to show you different sizes of field. This is for my small fields from 5 hectares up to 25 hectares. And then this is for my large fields, still having 25 hectares and then going up to 200 hectares. And what's cool is that we see that the fastest predators experience the greatest loss of control. So kind of as I move from left to right, the increase in aphids goes up. But this effect saturates for very fast predators in smaller fields. So kind of they're already effective enough. So being affected by the spray doesn't make them worse at hunting the aphids or not noticeably. And we also see in very large fields, there's not so much of a penalty because of the pesticide spray. And in these cases, it's because the predator was already very ineffective as a biological control agent. So making it worse isn't having much of an effect on your outcomes. And then most recently, what we've done is try to think about more complex landscapes. I did have to switch to a 1D version of my PDE just because there was a lot happening. But in agricultural fields, managers can use trap crops that are to lure the pests away from the profitable principal crop. And it might also lure predators away from those crops as well. So this is kind of a diagram of the different scenarios we considered where bright green is the principal crop that you want to preserve and dark green is like a grid that you've devoted to trap to try and draw the pest away. And we thought about this in block configurations where you have a lot of trap and then a lot of principal, or in mixed configurations where you really tried to evenly distribute that trap crop so that it's nearby principal crops. And then just different percents of trap from 10% to 50%. The movement between these like grids is going to depend on how palatable the crop versus trap is. And so we just explored how the predators might respond differently and how that would affect pest control. What we did was we thought about increasing levels of palatability, sort of how attracted the pest is to the trap crop. And then a few different predator responses. So we thought about the case where the predator shares the pest's attraction to the crop, a case where the predator kind of followed but a little bit behind the pest's attraction to the crop, and then a case where the predator had its own preference for the crop independent of the aphid. And we looked at all these different scenarios and thought that the mixed high trap case would be the best, but contrary to expectations, mixed configurations were actually worse than blocked, just because predators who found the trap crops palatable would kind of go in there and not come out. We also found that predators who closely followed the pests would be worse than having no predators at all, just because they would continually reduce what's in the trap area, kind of allowing more from the crop to build up. And here's just a sort of graph of the blocked landscape with invariant movement. The predators had their own movement that they didn't depend on the pest preference to the traps. Just because that was the case where we saw the most effect of the predator community. And then along the horizontal axis is time, and along the vertical axis is average across all my crop grids. And my red lines describe the effect of having no trap crop, and my dashed versus solid lines describe having no predator versus a predator. And there is just this overall effect that predators reduce the pest abundance. They kind of bring down the maximum that we reach here, and trap crops slow the approach to that maximum, kind of moving from left to right between those red and yellow lines. So just takeaways were that fostering predators in small fields led to the most effective pest control outcomes. We saw a significant decrease in pest control with field size. And pesticide effects were largest in mid-sized fields and minimal for most predators in small fields, and sort of couldn't make the situation worse in large fields. And there's a non-intuitive interplay between palatability and trap use, but you can have them synergize in some scenarios, and we kind of want to look into this more in the future. Next steps are that I would like to, similar to my prior work, solve an optimization problem on this type of scenario, but I would need a more sophisticated numerical solution to the model. So I've been working with some numerical PD folks here at Texas Tech to do that. And then eventually we'd like to use the models to describe how external drivers and their changes could affect management choices and outcomes. So we could incorporate some kind of temperature dependence or the effect of like neighboring land development. So I think that this modeling approach could be useful for just discovering how agricultural choices could change with new conditions. Right now, what we're also trying to do is compare some different modeling approaches to sort of encourage their use in this area of research. My collaborator on this work is like working on a book chapter related to trap cropping. So we're comparing differential equations, just compartmental, the current work with PDs, and then also some agent-based models. So with that, I just want to say thank you to you all for having me here, and I'm happy to answer any questions.
Okay, great, thank you. That was a wonderful talk, really interesting. Just hitting the clapping emoji there if anybody else wants to do that too. So yeah, we definitely have some time for questions. If anybody has some, please go ahead.
Or discussion. I saw on the
yes.
webpage that it's for discussion time.
So, yeah, Hans, please go ahead. Oh, you might be muted though, I think.
Yes, I am. I was. Sorry. Thank you for a very, very interesting talk. Good application of mathematics in biology, right? So thank you very much.
Thanks.
What in your models? There's no effect of the wind.
Of the wind, no. Yeah.
I assume that will complicate the problem considerably. Have you thought of that?
So in like, oh, let me go back. Where did I show you this? Wherever I showed you that predator community, this one, in this work with Kirchstadter, we saw these like odd drops in the aphid population sometimes that biologists told us was probably because of wind, like they get knocked off the plants. And it like made it harder for us to parameterize our model because it was a feature we didn't consider. So yes, I've thought about the fact that it's missing that, and I have not addressed it. It seems quite complicated to me and unpredictable.
Okay, thank you. Any
But
other
yeah.
questions? Sorry. Yes.
Oh, but just that's a really good point. Just not my area.
Okay.
I had kind of a hypothetical question when it comes to the temperature. So if one of the things that I think will be more common in the future is just sustained intense heat waves. And I wonder if you had one that lasted for a few weeks or something, and it drove the predators into, I guess, more of a lethargic state where they weren't actively hunting, could you get like a shock to the system where you'd have a boom of an aphid population that would then be hard to control again without a pesticide, or might damage the crops in the meantime?
Yeah, that definitely could happen. I do think like if the temperature was high enough, it would also kill the aphids a little bit, just because they're also sensitive to temperature. But like there's the sweet spot where the temperature would actually improve their growth and the predators would just be like knocked out. And in that case, yes, they could just sort of colonize. And then sort of a quirk of aphids is that once they're on a plant and they start to build up, it makes it easier for more to colonize the same plant. So just kind of it unleashes a problem that you can't take back.
Just as a slight follow-up to that, in growing tomatoes this year, I was introducing ladybugs right into my garden, and one of the things that I tried to do is keep them from flying away, and so it was trying to make an environment that they were happy in, right? So this is really relevant to summer activities, but I wonder if in that case, could you prep a little bit by providing some kind of refuge habitat for the predators so that they can kind of hunker down through it and then emerge to attack the aphids again?
Yeah, so I think I saw when I was doing the lit search for this an example in orchards where they tried that, sort of designing shelters nearby the plants that you want, sort of covered by predators, so that there's a very close by place that they can seek shelter and then return to the plant.
Okay.
Yeah. And like their finding was that yes, as you'd expect, that is beneficial.
Interesting. Cool.
In that case, would you get delayed differential equations? The predator going back to their home turf and then coming back later?
So the way that like I would describe it in my current modeling framework is that they would just have a time where they're not there. Like I only describe the pest's turf and then whether the predators are there or not. And then once the temperature was appropriate again, they would come back. I think you could think about delay differential equations, especially based on like how the current temperature isn't what matters, but like the past temperature that sort of drove them to take the behavior. Mine assumes it's instantaneous. And I think that leads to maybe not as complex dynamics you would have as if you accounted for that delay.
Okay.
So, hi, Amanda. How you doing?
Hi.
I'm good. You mentioned in your talk earlier that for the first part of your talk that you were thinking about extending your model. So have you thought about like what components or how would you try to do that? Like what directions?
Yeah, so kind of I had a laundry list of things that were missing from the model when we tried to fit it to the data. The most important of which is that this work only concerns predators, but parasitoids are actually quite important for control of aphid pests, like wasps that lay their eggs inside of aphids. So adding that would be like necessary next step. Additionally, the other types of things that could lead to large die-offs of aphids are things that I would like to include and sort of explore, like Hans brought up earlier, wind was something that had been proposed, or birds, But to be honest, I like insects, so I would probably do that last. And then also sort of something that is a limitation of the work that I showed today is that it all occurred over a single season. And to think about like long-term change in temperature, I would need sort of the compounding effect of over multiple seasons, your predator community changes and some flourish and some die off. And the thing that I can't do in my current model is account for when the predators reproduce. So sort of accounting for parasitoids would be my first thing. And then my second thing would be setting up a longer term model.
Mhm.
Thank you.
Yeah. And the longer term thing is hard because you have lots of like discrete pulses to agricultural
Yeah.
systems. So it's a really cool thing to think about.
Right.
If I may ask another question, out of curiosity, what is the largest system of ODEs that you're trying to solve?
So for the ODE one, it's like the 2, 3, 4, 5, 6, 7, 8, 9 predators and 1 pest. The parameterization that I used here before, I had to manually specify the like temperature dependent activity, but the body mass dependent thing, you can actually scale up to like arbitrarily large networks. It's quite a nice modeling framework. For the PDEs, I could not tell you like off the top of my head what size that system became after I discretized the space, but very large.
Did you ever run into the problem of stiffness? Depending on your parameters, obviously.
Yes.
So your parameters, they cover a wide range. Undoubtedly, you must have a problem with stiffness.
Yeah, it was really sensitive to these movement parameters could cause V to change like a lot very fast, and it would just kind of break the simulations. I'm not numerical enough to fix it. Like my rectangular grid approach was definitely not sufficient in those cases, and I didn't know how to fix it. So I just I limited it to the cases where that didn't happen.
I see.
Yeah, there was like a very small range of parameters here that I could explore.
Right. So there might be a possibility to do some asymptotic analysis if you have small various parameters that enter in a peculiar way, either being very small or very large. You might take advantage of that to reduce the size of your system and then somehow get an asymptotic picture that might explain what is happening.
Okay.
Just a suggestion.
Okay. I will probably write down that word and then Google it later. I've never done that before.
I see. Okay.
For a PDE. Like I understand for an ODE how it analyzed the asymptotic behavior, but
Yes.
using it to get rid of parameters in a PDE, I don't know what that is, but I'll look into it.
Yeah, that may be more complicated, obviously. But even in your ODE case, you said you had 9 or 10 species, and how many parameters?
So it's like the parameters are on the order of like n squared for n species,
Yeah.
but it's actually all written in terms of like 5 scaling parameters. And then the rest is calculated based on their body mass. So it's a very fun parameterization. It's not as many parameters as you'd think from looking at it.
Right, right. Okay.
I'm sorry, I got a question. I missed something with the directed movement, the V. So that's like wind or something, or?
No, it's the prey taxis of the predators. Like they can sense not necessarily the pest, but a plant that's being negatively affected by the pest, and then they're drawn in that direction because they know that's where their food source is. So it depends on the gradient of the pest to draw them in that direction.
Okay.
It's sort of an attraction factor, right?
Yes.
Okay. Oh, that's good.
Okay. Are there any more questions? I see Christian stepped away.
Thank you.
Yeah,
Thank you very much. That was great.
Yes.
yeah, thanks for having me. You all like had such a detailed discussion. I'm really glad I had the 15 minutes for it.
Yeah. It's just a great example of how mathematics can be used in so many wonderful situations to optimize and plan and considering temperature and how predators might be affected. And it's really, yeah, this is really cool stuff.
Mhm.
Thanks.
Okay, yeah, I guess if there's no more questions, let's thank our speaker again. I'll click on clap and clap on the camera. And thank you very much. I guess, let's see, I think next week we do not have a talk. There was a cancellation, unfortunately, but we will pick up the week after that for sure. And hope to see you all then.
Mhm. Thank you.
Thank you.
Thank you.
Thanks, Amanda.